Vaishali, and I'm from Mangalore. Yes. Not Bangalore, Mangalore. Yes. Have you all got your material? Who's giving you the material? Where is Mandara madam? Yes? Do we have to wait for her till you get your material? No? Without the material, how are we going to solve? Only synopsis without the material. So we'll start with the molecular basis of inheritance. Molecular basis of inheritance. Yes. What have you all studied in this particular topic? Do you all remember? Yes. So you all have studied about nucleic acids, is it not? What are nucleic acids? Where are they present? They are present in the nucleus, is it not? That is why they are known as nucleic. Yes. They are found in the nucleus and they show acidic property. That is why acids. Who discovered them? Frederick Mister, right? Frederick Mister discovered nucleic acid and he saw this nucleic acid, he gave the name to this nucleic acid as nuclein. Nuclein anta hesranna kottru. The term nucleic acid, the word nucleic acid was given by Altman. Yes, Altman gave the term nucleic acid. So, two types of nucleic acids. Yes, one is the DNA and the second one is, right, the RNA. The first one is the DNA. So, what's the full form for DNA? Right, okay, deoxyribo, yes, nucleic acid, right. Now, where do we find them, students? Elli irodu? DNA. Yes, it is found in the nucleus, agreed. Other than the nucleus, where else do we find them? Nucleus alli matra iruda? Where else do we find them? Yes? No, not in the cytoplasm. They are found in the chloroplast. Remember last year when you all had drawn the chloroplast, inside the chloroplast you all have drawn DNA. So, it is found in the chloroplast. It is found in the mitochondria, right? In the mitochondria also DNA is present and it is also found in plasmid. What are plasmids? Extra chromosomal double stranded circular DNAs found in bacteria, right? So, in the nucleus you find them in the chloroplast, right? Yes, we find them in the mitochondria. Plasmid, not plasma membrane, plasmid, right? Yes. Now, what is the chemical composition of DNA? What is the DNA made up of? All of us know that DNA is negatively charged, is it not? Yes, okay. Now, what is the chemical composition of DNA? What is the DNA made up of? It is made up of pentose sugar, is it not? Yes. So, chemical composition includes so it is made up of pentose, right, pentose sugar. And which is the sugar present in DNA? Deoxyribose, right, deoxyribose, right, yes. And the molecular formula of deoxyribose is C5H10O4, one oxygen less than the ribose, deoxy, one oxygen less than ribose, right. So, C5H10O, yes, 4. So, this is the molecular formula, right, yes. Adallade, what else is present in the DNA? It is made up of pentose sugar. Second component present in the DNA is the nitrogenous base, right, nitrogenous base. So, there are two types of nitrogenous base, is it not? Which are they, students? Purines and, right, pyrimidines. So, purine, the first one is the purines. Now, purines, these are larger in size and they have got two rings. Idralli eradu ring untu. One hexose ring and the other one pentose ring, right. So, these are larger in size and they are double ring. One is the hexose ring and the other one is the pentose ring, right. Now, which are the two types of purines? Yes, adenine and guanine, right. Adenine represented as A and yes, guanine represented as G, okay. Pyrimidine, right, pyrimidine, this is smaller in size and it has got only one hexose ring, right. And the two types of pyrimidines include cytosine and thymine, right. Uracil in RNA, I am talking about the DNA, right. So, cytosine matte thymine, right. Cytosine represented as C, thymine as T, right. And which is the third component present in DNA? Moorne component yaudu? Yes? Phosphoric acid, right. Phosphoric acid. And the formula is H3PO4, H3PO4, right, yes. Now, pentose sugar, if you represent the pentose sugar, if you write the structure, this is how it looks, right. This is the first carbon atom, second, third, fourth, and here we have the fifth carbon atom, right. And then we have OH, H, right, H, yes, HOH, CH2OH and H, right. So, this is the pentose sugar present in DNA, right, okay. Now, what is a nucleoside? What is a nucleoside? Nucleoside andre entadu? It is a combination of pentose sugar and any one of the nitrogenous base. Pentose sugar plus a nitrogenous base gives a nucleoside. So it is made up of pentose sugar plus, yes, nitrogenous base, right? And the bond that is formed here, students, between the pentose sugar and the nitrogenous base, the bond that is formed is known as N-glycosidic linkage or bond, right? So ಇಲ್ಲಿ pentose sugar ಮತ್ತೆ nitrogenous base, any one of the nitrogenous base, it can be adenine, guanine, cytosine, thymine, ಯಾವುದೇ ಒಂದು nitrogenous base ಇರಬಹುದು. So the bond that is formed here is known as N-glycosidic bond. Yes. N-glycosidic bond. Now where is this nitrogenous base attaching to the pentose sugar? To which carbon atom? Yes. Nitrogenous base ಯಾವ ಕಾರ್ಬನ್ ಆಟಮಿಗೆ ಅಟ್ಯಾಚ್ ಆಗ್ತದೆ? Molecular basis of enhance. ಯಾವ ಕಾರ್ಬನ್ ಆಟಮಿಗೆ ಅಟ್ಯಾಚ್ ಆಗ್ತದೆ? It attaches to the first carbon atom. So here what is present, students? Nitrogenous base. So nitrogenous base is attached to the first carbon atom. And what is the bond formed between a sugar and the nitrogenous base? It is N-glycosidic bond, right? Yes. What is a nucleotide then? Nucleotide is made up of all three: pentose sugar, nitrogenous base and phosphoric acid, right? Pentose sugar, nitrogenous base plus phosphoric acid. Yes. So what is pentose sugar plus nitrogenous base? So can we write this as nucleoside plus phosphoric acid? Yes, yes. So this is nothing but nucleoside, yes, plus phosphoric acid, right? And the bond that is formed between a nucleoside and a phosphoric acid is phosphoester bond. The bond that is formed here is called ester bond. ಈ ಬಾಂಡ್ ಅನ್ನ phosphoester bond ಅಂತ ಹೇಳೋದು, right? And phosphoric acid is attached to which carbon atom of the sugar? Fifth carbon atom. So here what is present? Phosphate group or phosphoric acid is present. Understood? And ಇದರ ನಡುವೆ ಫಾರ್ಮ್ ಆಗುವ ಬಾಂಡ್ ಯಾವುದು? Phosphoester bond. So first carbon atom, sugar is the first carbon atom ಗೆ what is attached? Nitrogenous base. Fifth carbon atom ಗೆ what is attached? Phosphate group. Phosphoric acid. Agreed. Okay. Third carbon atom ಗೆ what is attached? OH group. Right? OH group. Remember this? First carbon atom ಗೆ ಏನು attach ಆಗಿದೆ? Nitrogenous base. Fifth carbon atom ಗೆ phosphoric acid. And third carbon atom ಗೆ OH group. Right? Yes. Now what is a polynucleotide? Polynucleotide ಅಂದ್ರೆ ಏನು? Many nucleotides join. ಇಂಥದ್ದು ತುಂಬ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ಗಳು ಜಾಯಿನ್ ಆದಾಗ ಎಂತ ಸಿಗ್ತದೆ? Polynucleotide is formed. Okay, so this is formed by the joining of many nucleotides. And what is the bond that is formed between the two nucleotides? Yes? No? Not hydrogen bond. Yes? Phosphodiester bond. Yes, good. So the bond that is formed between the two nucleotides, this bond is known as the phosphodiester bond, right? Phosphodiester bond. So ಎರಡು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಗಳು ಲಿಂಕ್ ಆಗೋದು ಎಲ್ಲಿ? Now this is one nucleotide, right? Now suppose if I write the same structure again here. So I am going to write the same structure here, right? Numbering 1, 2, 3, 4, and this is the 5th carbon atom, right? Yes. What is present in the first carbon atom? Okay, nitrogenous base, right? Here you have H and H. Here you have H, OH. Okay. Here we have CH2, OH, and here what do we have? Phosphate group. Okay. Now the bond. Nucleotides, ಎರಡು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಗಳ ನಡುವೆ the bond that is formed ಯಾವುದು? Phosphodiester bond. And which carbon atoms are involved in the bond formation? It is the 3rd carbon atom of one nucleotide and the 5th carbon atom of another nucleotide. Bond formation ಅಲ್ಲಿ ಯಾವ carbon atoms involve ಆಗಿದೆ? 3rd carbon atom of, so illi bond form ಆಗ್ತದೆ, okay? ಈ ತರ, right? So the carbon atom that is involved in the formation of bond is 3rd carbon atom of one nucleotide and the 5th carbon atom of another nucleotide. Yes. ನೀರು ಇಲ್ಲಿ ನೀರು ಸಿಗ್ತದೆ ಮ್ಯಾಮ್? ಉಂಟಾ ನೀರು ಉಂಟಾ? Okay, you'll have your books, you'll have your, okay, material. Okay, let's look into the first question. See this. This you all have studied under packaging of DNA, right? Now the DNA is so huge. Is it not? Now suppose if you remove the DNA from one cell, ಒಂದೇ ಒಂದು ಸೆಲ್ ಇದು DNA ಅನ್ನು ನೀವು ತೆಗೆದು ಬಿಟ್ರೆ, and if you unwind it, ನೀವು ಅದನ್ನು ಓಪನ್ ಮಾಡಿದ್ರೆ, you know, it is as tall as you are. ನಿಮ್ಮಷ್ಟು ಹೈಟ್ ಇದೆ. Just, you know, the DNA of one single cell. Now such huge DNA, how is it placed inside the cell? Cell is not visible to our eyes. ಸೆಲ್ಲೇ ಕಾಣುವುದಿಲ್ಲ ಅಲ್ವಾ? ಆ ಸೆಲ್ ಒಳಗಡೆ, ಅಷ್ಟು ಚಿಕ್ಕ ಸೆಲ್ ಒಳಗಡೆ ಅಷ್ಟು ಉದ್ದ DNA is present. Now how is it placed there? By packaging. Now we know that DNA is negatively charged. Is it not? DNA is negatively charged. And DNA, this DNA is wrapped around positively charged histone proteins, right? It is wrapped around positively charged histone protein. Now histone proteins are of 5 types. You must have studied. There are 5 types of histone proteins. Which are they? H2A, H2B, H3, H4, right? So there are 4 types of, actually 5 types, is it not? 5 types of histone proteins, right? They are H2A, H2B, H3, H4, and there's one more, that is H1, right? H2A, H2B, H3, H4 ಮತ್ತೆ H1. Now H2A and H2B is rich in amino acid lysine, right? It is rich in amino acid lysine. H1 is also rich in amino acid lysine, whereas H3 and H4, these two are rich in amino acid arginine, right? Yes. Now these, no, each one of these are present 2 in number. Prati undu kuda it is present 2 in number. Idu onde irodu, right? So idanna ನಾವು draw ಮಾಡಿದ್ರೆ this is how it looks, right? This is how it looks. So H2A, H2B, H3, H4, and I said each one is present 2 in number, right? So H2A eradu itara, H2B is also 2, H3 is also 2, H4 is also 2, right? And this results in the formation of an octamer. 8 idiyalla illi, so this results in the formation of an octamer. So this is histone. Histones are positively charged. Around the histone protein, what is getting wrapped? Yes, DNA is getting wrapped like this. So DNA is negatively charged, histone protein is positively charged, right? So this is an octamer. After wrapping the extra DNA, you know, it comes out like this, right? Now the two nucleosomes or the two octamers, ee tara eradu octamers idiyalla, they are linked to each other. ಇವುಗಳು ಲಿಂಕ್ ಆಗಿದೆ, ದೇ ಆರ್ ಕನೆಕ್ಟೆಡ್ ಟು ಈಚ್ ಅದರ್ ಬೈ ಒನ್ ಮೋರ್ DNA. ದಿಸ್ DNA ಈ ಹೊರಗಡೆ ಬಂದಿದೆಯಲ್ಲ, ದಿಸ್ ಇಸ್ ದ ಲಿಂಕರ್ DNA ಆರ್ ದಿಸ್ ಇಸ್ ದ ಸ್ಪೇಸರ್ DNA, and in between this you can see the protein that is present. This is H1 histone protein. ಇಲ್ಲಿ ಇರೋದು H2A, H2B, H3, H4, right? ನಂತರ ಎರಡು ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ಸ್ ಅಥವಾ ಎರಡು ಆಕ್ಟೋಮರ್ಸ್ ಅನ್ನ ಕನೆಕ್ಟ್ ಮಾಡೋದು ಯಾವುದು? ಲಿಂಕರ್ DNA ಅಥವಾ ಸ್ಪೇಸರ್ DNA. ಅದರ ನಡುವೆ ಇರುವ ಪ್ರೋಟೀನ್ ಈಸ್ H1 protein, right? Yes. Now this structure ನಿಮ್ಮ ಬುಕ್ ಅಲ್ಲಿ ಹೇಗೆ ಕೊಟ್ಟಿದ್ದಾರೆ ಇದನ್ನ, this entire structure ಇದನ್ನ ಒಂದು ಬಾಲ್ ತರ ಕೊಟ್ಟಿದ್ದಾರೆ, ಕರೆಕ್ಟಾ? So this is histone octamer, right? And then yes, DNA is, this is the DNA, correcta? This is the DNA. Identify the right option. Right, option C, right? Okay. Okay, next moving on to the second question. Look into the second question. Match it, students. Is it possible for you all to match? Griffith transformation. Remember studying Griffith's experiment? What did he do? Right. He took the mice and he worked on which bacteria? Streptococcus pneumoniae. ಎರಡು ಸ್ಟ್ರೈನ್ಸ್ ಅನ್ನ ತಗೊಂಡ್ರು, ಸ್ಟ್ರೆಪ್ಟೋಕೋಕಸ್ ನಿಮೋನಿಯದು R strain ಮತ್ತೆ S strain, and he injected this to the mice, right? So you all remember that experiment, okay? Yes. Hershey and Chase. Transduction, okay. Measles and Stahl. Semi-conservative method of DNA replication, right? Jacob and Monod. Lac-Operon concept. Okay, option A. Which of the following phenomenon was experimentally proved by Meselson and Stahl? Do you all remember that experiment? Semi-conservative method experiment. ನೆನ್ಪುಂಟಾ? ಏನ್ ಮಾಡಿದ್ರು? Meselson ಮತ್ತು Stahl ಅವರು ಏನ್ ಮಾಡಿದ್ರು? Yes, students, what did they do? They worked on E. coli. Bacteriophage alla, they worked on E. coli, right? So what they did was they grew E. coli cells anna elli grow madidru. They grew in a medium containing ammonium chloride, right? They grew E. coli cells in a medium containing ammonium chloride, and the nitrogen of this ammonium chloride, ಈ ಅಮೋನಿಯಂ ಕ್ಲೋರೈಡ್ ಇದು ನೈಟ್ರೋಜನ್ ಉಂಟಲ್ಲ, this was radio labeled, right? It was made heavy. So what happened? This E. coli cells, no, they were grown in this medium for several generations, not for 1 or 2 generations. They were grown in this medium for several generations. So what happened? Nitrogenous base contains DNA ಅಲ್ಲಿರುವ nitrogenous base ಅಲ್ಲಿ ಎಂತ ಉಂಟು, ನೈಟ್ರೋಜನ್ ಉಂಟು ಅಲ್ವಾ. ಸೊ ಈ ಹೆವಿ ನೈಟ್ರೋಜನ್ ಹೋಗಿ ಎಲ್ಲಿಗೆ ಬೈಂಡ್ ಆಗ್ತದೆ, DNA ನೈಟ್ರೋಜಿನಸ್ ಬೇಸಿಗೆ ಬೈಂಡ್ ಆಗ್ತದೆ, right? So what happens, both the strands of DNA becomes heavy, N15. ಎರಡು ಸ್ಟ್ರಾಂಡ್ ಅಲ್ಲಿ ಕೂಡ ಏನಿರ್ತದೆ, N15 will be present for several generation. E. coli cells ಅನ್ನ ಎಲ್ಲಿ ಗ್ರೋ ಮಾಡ್ತಾರೆ, ಇದರ ಮೇಲೆ ಸುಮಾರು ಕ್ವೆಶ್ಚನ್ಸ್ ಬಂದಿದೆ. That is the reason why I am explaining this concept. Okay, so initially what did they do? They grew E. coli cells in ammonium chloride, where the nitrogen of ammonium chloride is made heavy, that is, it is radio labeled, it is made N15, right? So what happened, both the strands of DNA are incorporated with N15. ಇದನ್ನ ಹೇಗೆ ಕಂಡುಹಿಡಿದ್ರು? Now the DNA of E. coli cells were subjected to density gradient centrifugation using cesium chloride, right? ಈ DNA ಅನ್ನ ಇವ್ರಿಗೆ ಗೊತ್ತಾಗಬೇಕಲ್ಲ, ಎರಡು ಸ್ಟ್ರಾಂಡ್ ಅಲ್ಲಿ ಕೂಡ N15 ಉಂಟು ಅಂತ. So how did they found out this? By subjecting the DNA to density gradient centrifugation using cesium chloride. Yes. So DNA was subjected to density gradient centrifugation using cesium chloride, and the DNA got collected. ಈ DNA ಎಲ್ಲಿ collect ಆಯಿತು? At the bottom of the test tube, indicating that it is heavy. So the DNA got collected at the bottom of the test tube. So this DNA is heavy DNA, right? Yes. Next what they did was, yes, they cultured these E. coli cells in ammonium chloride, but this time the nitrogen of ammonium chloride was not made heavy, it was N14, right? So E. coli cells anna next elli grow madidru, N14 medium alli grow madidru, ammonium chloride alli, where the N is N14. And after how many minutes did they get the next generation? After 20 minutes, yes. So after 20 minutes, they got the next generation. So what happened? These two strands unwind, correct? ಎರಡು strands unwind ಆಗ್ತದೆ. So this is N15 and this is also N15. And the new strand that is getting synthesized, this strand will be N14 strand. Yes. The new strand that is getting synthesized is N14 strand. Now this DNA, the DNA of E. coli is subjected to which one? Density gradient centrifugation using cesium chloride. So they have to know, is it not? ಎರಡು ಸ್ಟ್ರಾಂಡ್ ಅಲ್ಲಿ N14 ಉಂಟಾ ಅಥವಾ N15 ಉಂಟಾ ಅಂತ ಅವ್ರಿಗೆ ಗೊತ್ತಾಗಬೇಕು. How did they find this? They found this by subjecting the DNA to density gradient centrifugation using cesium chloride, right? Now this time the DNA got collected slightly above. Bottom alli collect aglilla. DNA elli collect aitu? The band was obtained slightly above, indicating that this DNA was not as heavy as the previous one, right? And this DNA is known as, yes, hybrid, right? So this DNA is known as hybrid DNA. So they didn't stop their experiment here, they continued with their experiment, right? Again they grew these E. coli cells where? In ammonium chloride, where the nitrogen of the ammonium chloride was not made heavy, it was N14. So ಎಷ್ಟು ನಿಮಿಷ ಆದ ನಂತರ next generation ಸಿಕ್ಕಿತ್ತು ಇವರಿಗೆ? 40 minutes. 20 plus 20? Yes. So after 40 minutes, they got the next generation. So these two strands of DNA unwinds, right? One is N15 strand, another is N14, correct? One N15, matondu N14. The next, idu kuda unwind agtade, N15, N14, right? Now the new strand that is getting synthesized will have what? N15 or N14? N14, because it has grown where? In N14 medium. So it will synthesize what strand? N14 strand, right? So illi N14 strand synthesize ಆಗ್ತದೆ. So this is also N14 and this is also N14. Now you can see here 50% of the DNA, 50% of the DNA is hybrid DNA and the remaining 50% of the DNA is normal DNA, not heavy. Normal, heavy alli enide? N15 ಎರಡು ಕೂಡ. Here N14 normal DNA. How did they find this? By subjecting the DNA to marilikilla idanna density gradient centrifugation using cesium chloride, right? Yes. So idanna hege ಕಂಡು ಹಿಡಿದ್ರು? By subjecting the DNA to density gradient centrifugation using cesium chloride, and this time they got two bands, ಎರಡು bands ಸಿಕ್ಕಿತು. One band was at the same level as that of the previous, right? ಒಂದು band ಇದೆಯಲ್ಲ same level ಅಲ್ಲಿತ್ತು, indicating that it is hybrid DNA, and one more band was seen slightly above this, indicating that it is normal DNA. Yes, normal DNA. And from this it is confirmed that DNA replicates by what method? Semi-conservative method, right? Yes. So that is andrenu pentose sugar plus nitrogenous base, correct? Pentose sugar plus nitrogenous base is nucleoside. These are the nucleosides of RNA, students, not DNA. So RNA alli pentose sugar yaudu? Ribose, right? So ribose plus nitrogenous base adenine gives adenosine. The nucleoside is known as adenosine, right? And then they have given their thymidine. So ribose plus thymine or deoxyribose ಥೈಮಿನ್. DNA ಅಲ್ಲಿ deoxyribose plus thymine gives thymidine, right? Yes, so it is the fourth option, DNA basic molecule. No, it is the genetic. Yes. Next question. Given figure represents the DNA double helix model as proposed by Watson and Crick. Select the option that shows the correct measurements of A, B and C. Remember studying the structure of DNA? Who proposed the structure for DNA? James D. Watson and Francis H. C. Crick, based on the X-ray diffraction studies of. Two more scientists are involved here. Who are they? James D. Watson ಮತ್ತು Francis H. C. Crick ಅಲ್ಲದೆ there are two more scientists who are involved. Who are they? One is James D. Watson, as you all know. Francis H. C. Crick, right? And they gave the structure of DNA based on the X-ray studies, X-ray images that were obtained by two more scientists. And these two scientists are Rosalind Franklin and Maurice Wilkins, right? Based on the X-ray diffraction studies of Maurice Wilkins and Rosalind Franklin, right? And for this they were awarded Nobel Prize, is it not? So out of the four scientists, only 3 scientists received Nobel Prize. One did not receive. Rosalind Franklin, she did not receive because when Nobel Prize was awarded, she was dead. So she did not receive. So do you remember the diameter? Yes, the length of one turn and the distance between the two base pairs. Yes. A is 3.4 nanometer, it's in nanometer. B is distance between the two base pairs is 0.34 nanometer and C is 2 nanometer or 20 angstrom. Yes. Option which one? A. Yes. Right? Okay, next question. DNA is a polymer of nucleotides which are linked to each other by 3 prime 5 prime phosphodiester bond. ನೆನಪಿದೆಯಾ? ಸ್ಟಾರ್ಟಿಂಗಿಗೆ ನಾನು ಡ್ರಾ ಮಾಡುವಾಗ 2 ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಅನ್ನ ಹೇಗೆ ಜಾಯಿನ್ ಮಾಡಿದೆ? 3rd carbon atom of one nucleotide ಮತ್ತೆ 5th carbon atom of another nucleotide ಅಂತ ನಾನು ಜಾಯಿನ್ ಮಾಡಿದೆ. Yes. To prevent polymerization of nucleotides, which of the following modifications would you choose? Yes. Polymerization ಅನ್ನ prevent ಮಾಡಲಿಕ್ಕೆ ಯಾವ modifications, which of the following modification would you choose? ಎಲ್ಲಿಗೆ ಬಂದು the groups are attaching OH ಗ್ರೂಪ್ಗೆ ಅಲ್ವಾ? OH ಗ್ರೂಪ್ಗೆ ನೀವೆಲ್ಲ ಕಡೆಯೂ ನೋಡಿದರೆ ನಾನು OH ಗ್ರೂಪನ್ನು ರಿಮೂವ್ ಮಾಡಿ ಅಲ್ಲಿ ನೈಟ್ರೋಜಿನಸ್ ಬೇಸ್, ಫಾಸ್ಫಾರಿಕ್ ಆಸಿಡ್ ಮತ್ತು ನೆಕ್ಸ್ಟ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡನ್ನು ಲಿಂಕ್ ಮಾಡಿದ್ದೇನೆ. Is it not? So what's the answer? removal or replace of 3' OH group in deoxyribose. Yes. Chalk unta? Okay students, next question. DNA replication takes place at dash phase of the cell cycle. ಹೌದಾ? Interphase ಅಲ್ಲಿ last year you all have studied cell cycle and cell reproduction. ಅದರಲ್ಲಿ interphase ಅಲ್ಲಿ there are how many stages? G1, G2, S. G1, G2 ಮತ್ತೆ S phase. Correcta? So replication of DNA ಮತ್ತೆ duplication of centrioles occur during S phase of interphase of cell cycle. G1, G2 alla. Gap 1, gap 2 alla. It happens during S phase, right? If a double-stranded DNA has 20% cytosine, what will be the percentage of adenine in it? Now you all have studied Erwin-Chergaff's rule. Remember studying Chergaff's rule of base equivalence, right? So Erwin Chergaff ಅವರು ಒಂದು ರೂಲ್ ಅನ್ನ ಪ್ರಪೋಸ್ ಮಾಡಿದ್ರು, ಕರೆಕ್ಟಾ? Erwin Chergaff, he proposed a rule called Chergaff's rule of base equivalence, right? Chergaff's rule of base equivalence. What does it say? Chergaff's rule of base equivalence. Yes, the total number of purines in a DNA is equal to the total number of or the amount of purine is equal to the amount of pyrimidine, right? So A plus G is equal to C plus T, right? So A plus G is equal to C plus T. So A plus G by C plus T is equal to a constant, right? It is nothing but is equal to a constant, and this was given by Erwin Chergaff. Now suppose if there are 20 adenine pairs with which one? Thymine. Adenine pairs with thymine, right? And between adenine and thymine, how many hydrogen bonds are present? 2 hydrogen bonds, right? Now suppose if there are 20 adenine, how many thymines will be there? There will be 20 thymines, correct? And cytosine pairs with guanine, correct? Cytosine ಯಾವುದರ ಜೊತೆ ಪೇರ್ ಮಾಡ್ತದೆ? guanine ಜೊತೆ ಪೇರ್ ಮಾಡ್ತದೆ. and cytosine ಮತ್ತೆ guanine ನಡುವೆ ಎಷ್ಟು ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ಸ್ ಗಳು ಇರ್ತದೆ? yes, there are 3 hydrogen bonds, right? So ಇಲ್ಲಿ 20 ಇದ್ರೆ ಇದು this has to be 50 equals 50. Remember, always it has to be 50 equals 50. So guanine ಎಷ್ಟಿರ್ತದೆ? 30. cytosine 30. So ಇಲ್ಲಿ ನಿಮಗೆ ಎಷ್ಟು ಕೊಟ್ಟಿದ್ದಾರೆ? 20% of cytosine adenine ಎಷ್ಟು ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ.